Katy O'Connell is our next speaker, and she will be kind of anchoring our tools and resources at the tail end here by talking about Zebrafish as tools. And then, of course, later in the day, we'll hear more about this from John Postlewaite. So please, Katy, take it away.
Thanks. It's on? Yes? Okay, great. So switching things up a little bit. I'm a veterinarian here. I work on Tucson campus as well as on the Phoenix campus, taking care of all the animals that we use in research. So my goal today is to just briefly introduce to you how zebrafish can be used in modeling human diseases, in precision medicine, and coming up with therapeutic solutions. So kind of in that One Health scenario. Why zebrafish? This is data from PubMed showing annual publications in zebrafish. You can see the trend here. They've been increasing their use in the past 20 years. Despite this, we only have one researcher at the U of A that's working with zebrafish. And they're working with larval zebrafish, which you can see in the bottom picture, are really easy to raise. They're just in a petri dish. They're small. This can be kept in your lab space. Whereas adult zebrafish, they do require a bit more sophisticated housing. There's different tank systems available. What's shown here is a recirculating tank system that our university animal care department does own. So we are able to support if more people want to work with zebrafish. And it's kind of my end goal here, right, is to get you excited and wanting to work with zebrafish so we can help support you in your missions. So if you haven't seen zebrafish before, this is them. They're tiny striped tropical fish. They live about 3 to 5 years. So a little bit longer than mice, which is our predominantly used species in research. But like mice, they are vertebrate species, and they have similar embryonic development to humans as well as gene homologous to humans, so how they can be used in translatable research. Why zebrafish? Several benefits to why we use them in research. First being, since they are so small, we can house thousands of them in one room. Even more so than we can with mice, so this makes them pretty cheap on your end for working with them. And on the kind of regulatory side, they are lower on the phylogenetic scale compared to mice, so this fulfills our requirement to use the lowest species in research. They also reproduce at a very high rate, so within one day, one mating, you can get over 200 eggs from one female. So when you have a breeding tank like this one set up here, you move the divider, the males and females do their thing, the eggs are fertilized externally, they drop through the filter, you collect them at the bottom, you can have thousands of eggs in one day to work with, fertilized embryos to work with. And the embryos, like I said, they're fertilized externally, they're also transparent, they're tiny, you can work with them under a microscope. So this is images showing the development at different time points. Their development is standardized, so at a set temperature, you can know at different points in time exactly where they should be in their cycle. And within 72 hours, they're in their larval form. By 5 days, they're swimming and eating on their own. So rapid development that allows you to do a lot with this. One of the things you can do, since we know how the development proceeds over time, is you can introduce these embryos to different chemicals and then study the changes. So this picture here on the left is your control. The other two rows are showing you different exposures to concentrations of chemicals and the changes in the phenotypes you can see shown here. Other things that you can do with the embryos, genetically modify them. So introduce fluorescent proteins, DNA. You can see here 2 genetically modified strains. So the one on the left has a pigment change, so loss of melanin. Homozygous at the top, heterozygous, the arrow is pointing to a little bit of melanin left, and then an ocular phenotype. Zerk at the bottom, Zebrafish Information Resource Center, has over 44,000 mutant strains that are already available for you to work with. One of the big developments and reasons people are enjoying to work with zebrafish is that they can be used in high throughput screening. This is a 96 well plate. Since they're so tiny, you can put one embryo in each well and then do imaging studies, expose them to chemicals, genetically modify them, and study it over time. Final benefit I'm going to talk about with you today is that they can be used in behavior studies. So they have a characterized behavior repertoire similar to like what we would do in our rodent species. All right, so how can you use them in precision medicine? Trying to have therapeutics that are directly tied to that patient, you want that rapid turnaround, right? So we talked about the high throughput screening. You can rapidly determine what therapeutics will work best for your patient. Also, since they can be genetically modified, you can study the genetic and molecular components that created the disease of interest. So this diagram just shows you all the different ways in which zebrafish are being used to impact precision medicine. Cancer therapy as well, cancer research. So zebrafish, they can be, I kind of mentioned, they can be introduced with genetic fluorescent protein. So that's what this picture here is showing. To study metastasis, you can also look at like vascular research with this. So this picture here, the arrows and the red is showing metastasis of a tumor, either in the untreated control or the treated on the bottom. So you can easily image them and study, cancer research in vivo. One of the drawbacks to using zebrafish was that they have a lower incidence and a later onset of tumors in comparison to mice. This is starting to be overcome, though. They have developed immune-compromised zebrafish that they've showed has similar growth rates, histology, and response to therapy as the NSG mouse, which is very commonly used in cancer research. So this is examples of the immune-compromised fish here. It's also transparent as an adult, so they can easily do imaging studies. And there are just other, not necessarily immune-compromised, but transparent as adults that can be used for imaging. Zebrafish, there are several models of infectious diseases with bacteria, viruses, but in immune research, the neat thing about zebrafish is their innate system is fully developed within the first few days of life, but their adaptive immune system isn't developed fully until around 4 weeks of age. So you can infect them at different time points in their development to study the innate system, either separate or combined with the adaptive system. Oh, and I should mention, microinjection is how they're commonly using the technique to inject into the embryos for genetic manipulation or infection. And now they have that automated, so it can be done in about 2,000 embryos per hour. So less man work for your technicians. Final part research that I'm going to discuss with you today is just the use of zebrafish in aging. So you can see kind of their characteristics here compared to mice and humans. High throughput screening is, again, very beneficial in the ability to genetically modify them here so that you can study the cellular and molecular components to aging in zebrafish. Previously, the high throughput screening was only able to be done in invertebrate species, so now we have a vertebrate model that this can be done in. So hopefully some of you are thinking about how you can use zebrafish, or you know other researchers and you can spread the word. But I'm here if you get the idea to use zebrafish and want to talk to us about how we can do it.
There was a paper just published last week. You could grow and titer the human norovirus in zebrafish, which is a big breakthrough because it's it doesn't grow in conventional cell culture. What's available is very difficult and very expensive. Do you have the ability to work with infectious human viruses like norovirus? I have a lot of stools, and it would be quite interesting to see if we can get them to grow in there because it's kind of a breakthrough if we can, because then you can evaluate water treatment and food treatment processes, which we were not able to do before.
Yeah.
So it's really great you have this available. But can you work with biosafety level 2?
We can make it work.
So you have a couple of those zebrafish systems available, and if we have researchers who want to use them, do you have spaces for those to go, rooms where they can go?
Always a hot commodity, spaces. We will make space. We do have, it may not be in the same building as where their lab is, depending on where their lab space is. So you might have to take the CAT train across campus, but we do have space on Tucson's facility where we can do it. We also have been told that there's space available for us to grow. We just need to get the budget plans for it, and then we'll be expanding.
Okay, and then a second thing that's relevant is part of our One Health initiative in building the fish lab. That's not a zebrafish lab, it's a cold water fish lab, and it has other kinds of advantages, and there's some disadvantages to them. But we did order an incubator that's available for researchers who wanted to work with zebrafish or stickleback or other fish models. That's on its way here, and so you can do controlled temperature and light experiments in petri dishes with fish embryos. That should be here soon, a few weeks or so.
Great. Yeah.
Okay.
No, I'm excited for that. Use Frank's fish lab, use our fish lab.
Yeah,
Here
so-
to support you.
So in the fish lab that we're developing for cold water fish, that'll be a stickleback fish lab. And for any of you who work on issues that have to do with sexual development, there are advantages to the stickleback model over the zebrafish model because stickleback have XY sex determination like mammals. And zebrafish, it's different and more complicated. John will talk a little bit about this, I think, later. And there's some things for which zebrafish will be better, but certainly for a lot of questions such as environmental chemicals that impact sexual development, stickleback are a great model for that. And then one other thing I wanted to mention around stickleback is that the stickleback family is the only family of fishes ever discovered to have an androgen-mediated protein. So they have a glue protein called spigin, which is the Swedish word for stickleback, and that is a protein that's produced in the kidney in response to high androgen levels because the male builds a nest and he glues it together with his kidney secretion. And you can do assays, therefore, if you have an environmental contaminant, for example, that is anti-androgenic, you might see if spigin levels are lower in males than they should be, or if it's androgenic, you can see if females are expressing this protein because they normally wouldn't. In the same way as when we're looking at estrogenic contaminants, you can look at vitellogenin expression, which is an egg yolk precursor protein. You look at that in the males because they shouldn't be making any egg yolk. Or if it's anti-estrogenic, you can see if the female expression layers are lower. And we also have good thyroid biomarkers and other things. So basically, it'd be a good idea to think about fish experiments, perhaps even comparing what you can do with zebrafish versus what you can do with stickleback. So we still have time for questions for Katy.
Briefly, Dr. O'Donnell, what does DACLAM, your added qualifications, stand for?
Diplomat of the American College of Laboratory Animal Medicine. So I'm a veterinarian, but I specialize in taking care of all the lab animals on campus. That was an easy question. You let me off the hook.
Other questions or just even brainstorming how you might want to use fish as a model organism?
So you said that there's a lot of these. Is it like a knockout model that's available of, zebrafish clones that you could just kind of order in?
Yes. Yeah. So Zerk has a bunch of them. Yes.
Okay, and then you could measure basically visually, like cell migration, angiogenesis, things like these.
Yep. Yeah. Just fluorescently tag them, and you can.
Okay. All right, and a immunocompromised fish model you could put human tumor cells into?
Yes. Yeah. That has definitely been done. And it
Yeah.
can be done.
Okay, interesting. Yeah, I think that'll be really useful.
Yeah.
Mice are expensive.
Yes.
Also, just to emphasize the scale of what you can do with fish in a small amount of space. Katie mentioned you can have thousands of animals in a small room. We actually did an experiment with stickleback embryos where we had 400,000 embryos in one room. So the scale is just completely different order than what you can do with mice and
Could you be so kind to let us know, can we use such a species as zebrafish as a food or feed? Human food or animal feed?
Feed zebrafish to humans or animals?
Yes.
You'd have to eat a lot of zebrafish. They're very tiny. But I'm not aware of them being used as food for
Like
others.
dry food or?
In India, it's a human food in parts of India.
All right.
You're good.
You rate them?
And similarly, although very few people eat stickleback, there are some places where stickleback are eaten. And my hometown is Anchorage, Alaska. There's a river that runs through the city called Ship Creek, but the indigenous name for the river means where the stickleback run, because the Tenina people who lived there, when there weren't salmon available, would make stickleback soup to eat. And you can catch them you can catch hundreds of thousands of them. Even though they're small, you can get them in huge numbers and have a nice fish meal as long as you can break down the spines, make a soup, something like that. So not a commonly done thing, but someone like Euphenia is working on creative ways around food security. Maybe that should be part of the solution. And lower trophic level fish, of course, have much lower levels of bioaccumulating contaminants such as mercury. So maybe it's sort of like getting into the world of eating insects. It might be part of our future.
Yeah.
Other questions for Katy? So we want everyone to be thinking, having their minds turning around use of fish models. We'll come back to it later today.